Roll some weed up here, man. Thank you, Steve, for providing an amazing booth with 2 employees to roll some cannabis for us, right? He's got some amazing jars out there of his finest stuff he's been reserving for us, so that's absolutely amazing. I did a podcast with Matthew Gates a long time ago, and he helped me straighten out some things because I was on this whole trip about how high bricks will keep bugs away, and I was on this whole trip about like, oh, I'm going to pull this refractometer out and squeeze all these leaves. I'm going to figure out all this information and this data, and realizing it was getting me nowhere. So Matthew Gates helped me with some few sobering moments, which was great in my podcast, and that's what I try to do with the Grassroots Living Soil podcast, is just bring out the rawest data possible and to selfishly help my own cultivation skills. And if it helps my cultivation skills, I'm sure it's going to help the listeners as well. So yeah, Steve, you want to tell us why it was so important to have Mr. Matthew Gates involved?
Yeah, I've been working with Matt for probably about 6 years. Hit him up, we hung out. How I work with people, I usually fly them out, we bro out, and we walk the space for a day, and we just hang out and we smoke. And I did that with Matt, and I thought maybe it'd help me, it would slow him down, and it would help me understand what the hell he's saying a little bit more. Smoking weed with this guy only makes him smarter, I'm telling you right now, dude. It doesn't slow him down at all. Anyways, long story short, this dude loves the industry, is definitely deeply invested into it, a wealth of knowledge. He's helped me on numerous occasions, whether it's misidentifying woodlouse hunters and catching brown recluse spiders, or dealing with the state in roly-polies. The state hates roly-polies, woodlouse. This guy's worked with me on writing SOPs just so people leave me alone about roly-polies and woodlouse. And I will be talking about that tomorrow because it is an issue for a lot of people. If you don't get it under control, they're going to start writing regulations that identify roly-polies, woodlouse as a pest, as a crop pest. So if you guys have overflowing roly-poly populations, please get it under control before it becomes my problem with regulations. But anyways, long story short, Matt's been a huge help with me and my team, so I hope you guys can really get benefit from this. He's got a different viewpoint on a lot of things, but I enjoy him, highly intelligent. Check it out, guys.
I really appreciate it. Hey, everyone. All right, cool, mic check. My name is Matthew Gates. I'm an integrated pest management specialist, although I feel like the phrase IPM specialist is kind of like HVAC technician, which I've told a few people here. I think that it conjures a feeling and a connotation that's not really what I'm about. It's about looking at a holistic, ecologically conscientious perspective, and I know a lot of the people here at the Living Soul Summit kind of already appreciate this, so I think you'll appreciate the point of this presentation, which I'll get into, but just a short thing about myself. I'm the founder of Zentinel Consulting. I do IPM support, pest management, things like Steve had mentioned already. You might know me from my YouTube channel, Zentinel, or you might know me on Instagram as @SyncAngel. I've written a few things about pests in cannabis in particular. One about cannabis viruses. I've contributed some chapters about arthropod pests in general, so insects and mites and things like this. And also generally about IPM theory. And that's a big part of what this presentation is about. So when I talk about holistic pest management, I like to use this concept called the Everswarm, which is this idea that the pests and also the plants and other organisms are constantly adapting, constantly changing. They've been doing this for hundreds of millions of years, right? And if we know this, a lot of times people are aware that like chemistries that people apply in conventional agriculture, if you apply them too often or the only thing that you apply is one particular mode of action, then various pests, they'll be selected for resistance in those populations. Then you can't use it anymore. But this is actually true for a lot of things, and it's not just true for chemistry. There are two major ways that this is, I think, easy to understand. One is there's like a genetic molecular level, so like things on the factor of like compounds you're applying, proteins and enzymes that are being secreted by biocontrols and microbes that you're applying, for example. So those are all molecules too, right? They're just bunches of them put together and they have certain effects. But that's like the small scale. And there's also kind of a larger scale. There are things that, and this is a contentious point, I think, but actually biocontrols can be resisted too. This is how evolution actually works in the real world, right? In nature. Various pests that we're familiar with, they will have behavioral reactions to things like the scent of their predator. If they detect the scent, then they might change how they deposit their eggs. They might move around more, and things like this. And although they're not necessarily easy to quantify in the field, it could be part of the reason why you might be having difficulty with biocontrols when you're utilizing them. And I think that these factors are important to consider, and there are things that you can do about them, which we'll get into. And also pests, of course, interface with cultivated land and also uncultivated land, and various pests that we get in, for example, the United States are not from here. Certainly some of the plants, or many of the plants that we grow in crops, are also not this way as well, and cannabis is no exception. It's not native to the United States or to North America or the Americas, as far as we can tell. And so that is actually going to be a factor in how you consider your ecological environment and how you are setting up your cultivation context. So I would say that the most important thing is that IPM has to constantly change to shape the pest adaptation through your own systemic or systematic actions. So how do you disrupt the everswarm? As a factor of evolution, you can't really stop it, but you can shape it. So much like the example with chemistries and things like this, if you understand that if you have a multi-layered or a multi-domain approach where you've got biocontrols, chemistries, and not just the same chemistry, but multiple kinds, things that are safe to use, things that are extracted from plant compounds, for example, things like this. If you have multiple layers, then you don't have very much selection pressure. So you have to look at three subjects mainly, I think. The pest physiology, which people who know my content know I love to talk about, get into the nitty-gritty, which can be a little bit technical, but I'm happy to answer any questions. So that would tell you things like their strengths and their capabilities, but also their weaknesses. And also their ecology, how do they interact with and how do they establish in different areas. Because even in native or even in natural environments where people are not cultivating plants, these invasive pests that get everywhere or they come from different parts of the globe, they can establish in these areas and then damage the environment and then damage your crops too. Not all pests are like this, but certainly there's a lot of examples. The big one that I like to mention is like chestnut blight, for example. It only took a few trees from Eastern Asia, bring them to North America, hundreds of millions of trees gone. Right? It didn't take very much for that to be a problem. And it's because those chestnut trees never had any experience with chestnut blight, right? So, and then their evolution. So if you understand how these pests evolve and adapt, if you know their reproductive cycles and things like this, and if they happen to be a pest that has a lot of history already, which some of the pests in cannabis do have this, like for example, rice root aphid was not a pest of cannabis first, it was a pest of, well, rice and many other crops too. So if you know how their physiology is, then you can take steps to avoid problems. This is a very busy graph, and I apologize if some of the text is a little bit wonky. The presentation, I had to hurriedly change like 60 slides here because the text was not the same for the presentation. So if you can't see it or read it or anything like that, feel free to ask me for a copy of the presentation. I'm happy to give it out for anyone, or if you have any questions. But this diagram is from a research report, and most of the research reports I cite in this presentation will be color-coded with the text, so you can see where I'm drawing this information from. But this shows an example of pesticide resistance in like a caterpillar as an example. But there's a bunch of factors that are also relevant to resistance to microbial pesticides and also resistance to plant defenses as well. So I recognize that it's a little bit hard to see, but one of those things would be like there are different receptors for compounds and toxins that the plants produce. And if those receptors change, then those compounds don't bind very well or at all with the receptor, and in that way, it will not function correctly. Insects also have an abundance of symbionts in their gut oftentimes that will also neutralize or suppress some of the compounds that are being produced in defense in the plant. They might also suppress the signaling of the immune system in the plant, and that kind of like stops there being a problem in the first place for the insect. And different populations will have different ways of being able to do this, not just for plants, but also for insects as well. And then there's a bunch of factors with regards to genetic detoxification and also how the expression of certain genes can change depending on the kinds of challenges that the immune system of the insect is dealing with, and vice versa with plants. This was a really cool research report. I'm taking some diagrams for it. This was about some oomycetes. So you might have learned about those a while ago or in some of the workshops that just came about. They're often thought of very similar to like fungi, right? But recent research has shown that they're actually descendants from like brown algae, if you can believe it. Their body structure is made out of cellulose, not chitin. So they're actually in some ways, they're a lot similar to very early plants than they are to like fungi, even though for a long time we thought that they had other traits very similar to fungi. And certainly functionally, that could be true for like pathogens. But the main point I want to elicit here is that pathogens, they have their own microbiome, and beneficial microbes, they also have a consortia of microbes that they work with. And what we found is that unfortunately you can't just say, which I've heard a lot of people say in various ways in the presentations currently or recently, is that there's no good guys and there's no real bad guys, right? And sometimes the good guys and the bad guys, quote unquote, pathogens and beneficials will work together in the soil microbiome to colonize the plant host. And in this diagram, this shows that on the upper left, this A section, there's various microbes, eukaryotic and prokaryotic microbes and viruses that are all associated with this pathogen. And then on the right, you have the host plant, and you have its cacophony of microbes that are associated with it, either closely or distantly. And on the bottom here, we have some oomycete spores. That's what the SP stands for here. And they're green, and that's because these are bacteria that are actually on the hull of the spore. And they're existing in tandem with the oomycete as it colonizes this plant host. So just something to keep in mind. This diagram I really like because it shows how plants have to deal with finite resources and how they decide to act towards pests. So on the side of A here, we have at the very bottom we have the resources. Obviously, plants have to make use of their finite resources. They have to acquire resources, and then those resources go on to fuel the expression of genes, right, and the development of tissues, various kinds and various ways. And then those tissues, of course, beget the traits that we're familiar with, some of which we can observe and other we can't really quantify without like sequencing them and that kind of thing. And then ultimately that there are selection pressures that then modulate whether or not those plants will go on to grow and reproduce, or will they fail in their environment. B here shows the growth defense hypothesis, or the theory of growth defense that ecological literature talks about, which is basically that when you have a plant growing, it has two options mainly for growth. It either puts its resources into vegetative growth, which we're all familiar with cannabis, and also reproductive growth as well. But it also has to put its resources into defense. Now, if it puts all of its resources into defeating the first pathogen or first pest that it detects, it's not going to have resources for growth. It's not going to have resources for other potential problems. So plants have gotten really good at proportionally reacting to problems that they interact with, essentially. And I promise that the pictures will be a little bit more interesting in the future. Dr. Surendra Dara, who is somebody I've associated with a lot, I really like his approach to IPM. And he talks about how there are other factors of IPM. A lot of people get hung up on the functional aspects, the practical aspects, like what chemical do I use, what biocontrol should I apply. But there's also, of course, economic and societal factors. Us as a people, us as a society, we decide as much as we can, what we think is acceptable, what we think is not acceptable, and I feel great camaraderie with the folks that I've talked to here at the Living Soul Summit that understands that that's a very nuanced conversation to have. But even if we feel that's important to work a certain way or to cultivate our plants a certain way, other people don't necessarily do it, and so we have to deal with the fallout of those decisions. This diagram, this shows in the section A here, most importantly, various areas that are likely to be invaded by invasive plants and animals. And we can see here that some of the major ones in the light blue color are North America, Southern South America, Northern and Western and Central Europe, Southeast Asia, Australia, and Southern Africa, and in particular South Africa, the country. And these are also really common places for trade. And trade is a major way that pests move across the earth. Obviously, a lot of people are familiar with, like, for example, Pangea, right? But even when all of the continents as we know them today were close together, I don't think it's been any safer or easier for various organisms, not just pests, but also the plants that we cultivate, to move across the earth really easily. And that has had a profound effect, and that's only been the last 200 years, which is a iota second of time in the evolutionary span. So as an example, this report showed the movement of one Connex box that started in the UK and then it came back to the UK. And in that time, 421 days, so over a year, it went 75,000 kilometers by ship, 5,000 kilometers by train, 2,000 kilometers by road, and it reached all of these major trade zones, right? Western and eastern coast of the US, Brazil, went past the Cape Horn, went into many important parts of Southeast Asia, 2 major ports in the People's Republic of China, Japan. It went through the Middle East, and then through the Strait of Gibraltar, and then back to the UK. So that's pretty insane to think. And all of these color codes talk about the amount of merchandise imports, and all these highlighted, the sort of robin's egg colored spaces are like trillions of dollars of money being exchanged in trade. 2 other major ways that pests move across the earth is through the cut flowers industry, which I have a lot of experience with like gerberas and roses and things like this, moving from South America in particular into North America and also areas of Europe and Africa. And also another one is subtropical and tropical forest deforestation happening in South America in particular, and also Africa, going into, in particular, Eastern Asia and Southeast Asia, or in Southern Asia, in, for example, India. These are major routes of invasive pest movement. And I'm bringing all this point to belabor that pest movement is egregiously simple, and that's why it's important to be on your toes. A few more points. Trade has increased quite a bit since 1992, and there's no real reason to expect that that's going to stop anytime soon. Both air and container freight has reached really high levels, and I don't even think the recent pandemic has changed that very much. Also, trade agreements have increased quite a bit, and we should also not expect this to change very much either. But there's a big correlation between alien species records, so invasivity and invasive species, and imports as a factor of GDP. And of course, North America and many other countries that we're familiar with are huge trade countries, and they're huge sites of invasivity. So I think that on a strategic level, on a communal level, IPM has to be, or it should be, local, it should be sustainable, it should be varied, and it should be rapid. You should react very quickly, and you should also be implementing preventative measures, which we'll go over. And also it should be something that other people can use. If it's a good system or if it's a good technique, but not everyone can use it, then it's not very revolutionary. So, and you want a layered resistance. Speaking of good decisions, though, we're here. This is where that arrow is. This is where Green Life Produce is, and you can see that Steve made a really great choice by by cultivating here, where the number of pests per county is pretty low. It's like about 3, it looks like, for this diagram. But you can see that, again, places that are close to the coast have a lot more pests per county in the United States. Western coast and also central, eastern, and northeastern United States are chock full of various invasive and exotic pests. Cannabis pests tend to have factors or traits that make them really likely to move into new places and be very effective and successful. Mostly that's going to be things like being able to reproduce often and constantly, and also high genetic diversity, which we'll get into with a few examples. But also a lot of places where we're growing are sites of disturbance, so basically any urban or suburban land, right? And also places where environmental stresses are increasing, whether it's due to a warming climate or whether it's due to environments just changing in general. That doesn't really bode well for the organisms that are already in the location to fend off these organisms. So that's the presentation, that's the intro. This is the Everswarm, Beyond Biodiversity for Holistic IPM. We're going to be going over 4 main pests, and we're going to talk about their capabilities. And this is kind of like a fun fact presentation, essentially, about what we're dealing with and what's perhaps not always appreciated about the pests that we're changing or challenging. So pest biodiversity is actually very vast and capable. I know we often talk about the biodiversity of the microbiome and other organisms that we like to facilitate, but it's also true of pests as well. So the first one is powdery mildew. Golovinomyces, which I think I remember Tad talking about in his presentation, and there are some others as well. This diagram on the left is a phylogenetic tree. It shows various fungi and their relationship at a genetic level, and also how related they are to other organisms. And we have this group called the Arachnopezzaceae, which I'm just going to call the spider puff, because that's what the name literally translates to. So the spider puff family, which is a funny name. And what's interesting about them is that the powdery mildews, or the Ericifiaceae, they descend from a common ancestor. And these spider puffs were saprophytic fungi. They were probably what we might consider beneficial fungi, and they still exist around now. And what they do is that they break down cellulose and they break down dead plant matter, essentially. And what we found is that powdery mildews, they share the same genes that allow them to do this. And what perhaps has happened is that over time, the basal powdery mildews, or the things that were powdery mildew before they were powdery mildew, perhaps they actually were like beneficial fungi, but then they developed more of an ability to exploit their host and then became parasitic instead of mutualistic, which is really interesting, I think. Especially since we often laud and lionize the importance of the soil microbiome, but sometimes it can bite back. So some of these similarities include that they share cellulose genes, but they also have shared communication pathways, which we'll go over. And they also fuse with the plant cell membrane, somewhat like some mycorrhizae do when they're infecting their host. Their genome is really big for ascomycetes or mold, other mold kind of fungi like aspergillus and that kind of a thing, generally speaking. They can move great distances. They've been tracked moving as much as 700 kilometers, which is fantastically large. And also, they can move across oceans. I have a video on my channel where I go over a new hazelnut powdery mildew. And in that research, they found 14 examples of powdery mildews that, and apparently this is very common, moved from North American continent into the Eurasian Western steppe and also into Asia across the ocean, which is really fantastic. And also one thing that I want to note is that powdery mildew spores, unlike the spores of a lot of other fungi, they actually contain water, or proportionally a lot of water in their spores compared to other fungi. And that actually allows them to develop in dry conditions. I know a lot of people control the environment, try to lower humidity for fungi. I'm not saying that's a bad thing or anything like that, but it might not be a perfect defense when we consider this capability. And also, when it comes to sexual reproduction with powdery mildew, when it does happen, they produce special spores that allow them to resist a lot of environmental factors like lots of cold and heat, aridity, and also wet weather. I like to say that for powdery mildew and other fungi, Every spore is a die roll. So what I mean by this is that, and I talked with a few of you here actually about this concept where they're obligate parasites. They can't develop on non-living plant matter. They have to develop on living plant matter that's a compatible host. And while a plant might complete its life cycle in the span of a whole year or a whole season if it's an annual, its pests, like these fungi, are going to be reproducing thousands of times in that period of time. And that allows them to adapt to their host a lot more quickly, if that makes sense. So if you're producing hundreds of thousands of spores, and maybe 95% of them, maybe some of them go off and they get onto the ground and they don't germinate and nothing happens, well, that's okay. But if the 5% are effective, then they're iterating on a development that's been happening for like 100+ million years. And those that are successful, well, they'll just go on to reproduce more themselves, right? So even if things are fine in your region, in your area, it might come to be the case that someday, sometime, some new super powdery mildew or some other pest will come in and potentially be a huge problem, even if you have all of your things dialed in. Another thing about powdery mildew is that they have what are called dynamic genomic compartments, which are basically these large sections of their genetic code that's basically turned towards constantly mutating and adapting, and these sections of genetic code are mostly composed of what are called virulence factors, which are all of the nasty compounds and toxins and things that they use to colonize the host, so all of their weapons. If you've never developed or never dealt with powdery mildew, I'm glad. I hope that never happens to you. But this is what powdery mildew looks like, and if you don't abate it, it'll look like a D over here. But it starts off as like a small white colony of like what looks like fuzzy hyphae, and then that expands rapidly. A lot of people think that powdery mildew is systemic, but it's not. We will talk about a systemic pathogen, and we'll compare the two, and we'll see why, in my opinion, systemic pathogens are way worse, like botrytis. But powdery mildews are not like that. They just exist on the surface of the plant for the most part. And they aren't typically lethal to their plant, but they do stunt growth quite a bit, very often. How does this happen? Well, a spore and the colonization of the plant is pretty invisible for like maybe the first 100 hours or so. It kind of depends. But they develop this structure called an aprosorium, which you can see in the diagram here. That will develop what's called a penetration peg, which is this physical, like a needle almost, that is coated in enzymes like the enzymes I mentioned earlier that break down the cellular structure. It basically melts through the plant cell. Cellulase and cutinase that goes through the waxy cuticle and that kind of thing. And that will eventually develop, if it's successful, into what's called a hostorium, which is like the major site of nutrient transfer from the plant into the powdery mildew. It's like a bulb. It's like a root, kind of. And then if that's successful, then the powdery mildew sporling will then produce more hyphae and then produce more aprosoria, and then the cycle will continue. I mentioned earlier that powdery mildews share a lot of, or at least some traits with some of the beneficial fungi we were talking about. We've known about these genes called mildew locus O genes in plants for a long time. They're shared between a lot of different plant families. And it's often been asked, when the MLO genes are not in their plants, if we've got mutants that don't have some of those, then the powdery mildew is actually not very capable of colonizing the host. Well, then why does the plant keep the MLO genes, right? Because if they got rid of them, or those that mutate without them, wouldn't they do better? Well, it turns out that MLO genes are necessary for mycorrhizal and other beneficial fungal interactions in the roots. So if you get rid of those MLO genes, then you get rid of the ability for the plant to interact with those mycorrhizae, unfortunately. And so what it is that powdery mildews exploit the same pathway that these fungi do. And that, again, I think lends credibility to the plausibility that they developed from beneficial or at least commensalic fungi that were helpful for plant development. So I think that's a really interesting aspect. And majorly, it's actually the plant cell's response to touch. or physical pressure. And so when it comes to like mycorrhizae or other fungi that will like fuse with the plant cell potentially, like arbuscular mycorrhizae do, plant or powdery mildew will use a similar kind of physical touch response and exploit that. When they do interact and when they do establish, powdery mildew are going to be constantly changing the expression of the plant cell. Everything or most everything in their genes are geared towards this manipulation of the plant host. There's not much, and reproduction, there's not much more after that. But plants have their own way to defend against this. They have two main times where they can deal with powdery mildew in sort of an elegant way. The first one is if they're kind of already primed against it, or if they happen to have what you might call an aggregate resistance factors that allow them to detect and also neutralize the powdery mildew, as we can see on the bottom right here. We call this an incompatible reaction. So the Apersorium tried to drill through, and it wasn't effective, and the plant cell lit it up with a bunch of toxins and also structures that will basically clog it and cause it to not be able to establish. If that doesn't happen, though, then the Haustorium is produced, and so what they have to do is they have to fight a big incursion that interferes with their ability to signal their defenses and also signal other cells, and they have to find a way to bind this Haustorium and not allow it to excrete all of these toxins and things. So this is the war that's going on when you get a powdery mildew problem. One thing that powdery mildew will do as well to fight this and to also enrich itself is that, believe it or not, it can actually cause individual plant cells to reproduce their genome, so become polyploid, but at a singular cellular level. It's thought that the reason why the powdery mildews do this is that it increases the amount of genes that are related to nutrient transport. So basically, it's making the plant cell produce factors that will increase the transport of sugars and other things that it wants. And even though you would think that ostensibly this would also increase the defense gene production of the plants, this doesn't seem to be a problem for the powdery mildew. So I think that's very interesting and kind of shows what we're dealing with here. But if the powdery mildew is to be destroyed, there's a two-part process for this to happen. And we'll see how this is exploited by other fungi, and I think it's really important to consider that when it comes to plants, just like us, plants have to deal with multiple kinds of defenses, multiple kinds of problems, and sometimes the solution to one problem is diametrically opposed to another. That's important because you can't do diametrically opposed things at the same time really well, right? You can't sit down and stand up at the same time. You can't look left and right at the same time, at least not without some kind of technical aid. And that's kind of what's happening here. So with powdery mildew, basically plants have a way to bind to the Haustorium, keep it from being able to excrete all of these nasty factors. And if they enclose it completely, then the the plant cell will destroy itself, programmed cell death. You may have heard of this. And they do this by increasing the ROS, the reactive oxygen species in the cell, and they basically destroy themselves, but they also destroy the powdery mildew. This is great, and you probably are aware that this is very common even in our own cells. This is a really common cellular defense mechanism. And if this is the first time the sporling is trying to develop on a plant cell, well then boom, it's dead. It's just like a seedling. It only has a certain amount of energy in its spore, and if it expended all of its energy and this gamble didn't work out, it's probably dead. And very briefly, I want to harp on this point that this has been happening for so long. So a little bit of evolutionary history, right? We understand, and the research is down here below, that powdery mildews developed about 100 million years ago on deciduous trees in the polar north when it was a lot warmer during the Cretaceous period. And that's what that green line signifies. So as those trees moved downwards, what would become one of the most important hosts for powdery mildew, the asters, So like sunflowers, or what would become sunflowers, and those kinds of things. Those plants are actually one of the most successful, or the most successful flowering plants. There's more aster species than other flowering plants. And those developed in southern South America. Then they moved into what would become Africa, North America, and Eurasia. And as they were moving northerly, the powdery mildew and its host were moving southerly, and they interacted, and then powdery mildews, or the basal ones, jumped onto these asters, and then the rest is history. Now there's a ton of powdery mildews that infect all kinds of flowering plant hosts. Another cool thing is that viruses are actually found pretty commonly in a lot of these powdery mildews, and it's only with recent technology that we've been able to kind of identify that that is the case. It's not like a commercial biocontrol or anything that's available, but it's interesting because I think that viruses, to be frank, are kind of slept on when it comes to the soil microbiome, and there's going to be some more examples of that in this presentation. And also just in the ecosystem at large. This is a Delta partitivvirus. It's not the same one that infects powdery mildews, but there are various ones out there. They cause a problem called hypovirulence. So powdery mildews that catch the virus, they're not able to colonize their host. And who knows, how often or to what degree this is happening all around us. I think that's happening a lot more commonly than we give it credit for. And I think that there might be ways that we can facilitate that in the future. So now I'm going to move on to botrytis, which is that diametrically opposed organism I was just talking about. So it's systemic. It actually exploits the defenses that we were just talking about to its benefit. And if you've never dealt with botrytis, I again hope you never do. But this is how the progress can start. This leaf next to the inflorescence in A, it becomes chlorotic, it becomes yellow, it becomes necrotic and brown as the infection moves through and up into the inflorescence, and then we see this sporulation that is very commonly called gray mold in botrytis. And there are other bud rot pathogens as well, but we're going to mostly talk about botrytis. Actually, botrytis cinerea. And botrytis cinerea is kind of a black sheep in its group of botrytis. Most botrytis species are actually specialists, believe it or not. They only infect one or a few different hosts that are closely related. But botrytis cinerea can infect like 1,400 plant species, which is egregious. And like I said earlier, it is systemic. It's even been found actually as a symbiont that does not harm its host in things like knapweed and also in like algae. So botrytis cinerea gets around, and a lot of people don't know that about it. And I think there's a lot of really interesting things to interpret about botrytis if it's able to have this wide gamut of interactions with various kinds of plants, even very early or primitive plants, as we would call it. And I think that's the most important thing. I think the other thing I want to say about botrytis is that they cause a lot of destruction and damage, of course. But with the right kind of layered defense, I think that it's much less of a problem than maybe people sometimes encounter. So like I said earlier, biotrophs like powdery mildew, they infect like the epidermal cells, and they create that hostorium. They kind of live on the surface. Botrytis and its kin, what they do is they very surreptitiously move through the cells. If it's a spore, sometimes they're vectored from their parents to their offspring, if you can believe it. So that does sometimes happen. But in the traditional model where a spore lands on the host, it'll germinate. The hyphae will move through the epidermal cells, and it will suppress the immune system around and inside the cells. And then insidiously, it will then cause the plant cells to kill themselves, program cell death, and then they'll sop up all the nice nutrients, and then they'll go on to do that on more cells, and they kind of rinse, wash, repeat that process. They do this a number of ways. They have a lot of things going for them. I'll talk about a few of those capabilities here. One of the things that they can change is they can change the delicate balance of reactive oxygen species in the plant cell. This is obviously going to really damage the plant if they hike it up or if they lower it. It's ROS can be a lot of things for plants. It's obviously a stress-related group, but also It can be like a signal in and of itself for the immune system. So they can modify that at will. They can also modify the delicate pH in the plant cell. This will have major effects on things like enzymes and proteins, and it might even neutralize things that are meant to damage the botrytis. And they'll also deploy effectors that will have many effects. It'll damage the plant cell. It'll change the genetic expression, much like we were talking about earlier with the powdery mildew. And if the botrytis gets some of these compounds or defense compounds that the plant cell produces, it's not a big problem, or at least for some of them, because they have these things called efflux transporters, which will allow the compounds to quickly move into and then out of the fungus. So they're actually able to deal with some of these plant defenses. So even a plant that has a lot of nutrient uptake and a lot of really important fortifications for its health, it can still have problems because botrytis is adapted to that. I really like this picture because it shows this sort of insidious hypha kind of meandering around the plant cell, and it shows some of these various effects, one of which are these things called small RNAs, or basically they're lines of genetic code, really small lines, and they will bind with the genetic code in the plant cell, and it's like kind of like putting a monkey wrench into a gearbox. It will stop the expression of that gene. So whatever that gene was supposed to become or do, it's no longer doing that because of this little monkey wrench that binds with it and stops the expression process. I think that's very sophisticated and elegant and sort of deserves our attention. But the major thing is that it can signal the programmed cell death process, and it can do that from without even infecting the cell intracellularly. It can just put out these effectors, these molecules. They'll bind with the receptors outside. That's what that C-shaped thing at the top is. And basically tell the plant cell to destroy itself. And then it just takes up the nutrients. It doesn't have to do very much to do that. These are various compounds associated with botrytis and other plant fungi. Do you see a theme here? Botryoidal, botrylendiol, dihydrobotryoidal, botryonolol, botryoic acid, botsinin. Yeah, there's a lot of compounds that are associated and were first discovered with botrytis. There's a huge arsenal of compounds. I'm not going to go through all of them because that would be a little bit too much. But the major one I want to talk about is in this light blue here, the first one called HIP-1. This stands for hypersensitive response inducing protein 1. And this is the major factor that botrytis and some other fungi are able to use. Or I think actually mostly botrytis, excuse me. But other fungi have some of these other arsenals at their disposal. But HIP-1 I think is specific to or is mostly associated with botrytis. And what it does is it causes that programmed cell death that I was talking about earlier. I like to call it plantcelldeath.exe for all of my computer friends here, because that's basically what it is. What research has shown is that if you deny botrytis and all of these other great factors I was talking about, and you just give it HIP-1, or you give a plant cell HIP-1, you expose it, that's it. The plant cell will destroy itself. It doesn't need any of the other things that we were just talking about. So all of that other stuff is gravy. And the HIP-1 is like kind of the major kill factor. And once they developed that, maybe it wasn't the first thing they developed in their evolutionary travels, but it's the most important one. And I think it just speaks to how fragile sometimes some of these defenses that plants have are. And the real big point I'm trying to make here is that even our own swords and our own defenses can be put towards ourselves if we're not aware of how they function at a very minute sphere. But not a lot of doom and gloom, right? We have other things that we can do about botrytis. As we can see, there are microbial biocontrols, and there are other organisms in the greater ecology that we can perhaps rely on. Maybe not in a really focused way. Sometimes we have to rely on commercial products, right? But these are 3 examples: Trichoderma asperellum, Bacillus amyloliquefaciens, and Gliocladium catinulatum. And compared to the control, which is highlighted this green A section, B, C, and D did pretty good in comparison. And a lot of times, in this case, they were applied 2 days before inoculation with the pathogen in this research. And you can see that they had a great effect. And going back to the Everswarm theory that I was talking about and how you disrupt it, a major way you do that, and this isn't new or novel. What I'm talking about has actually been known for a long time, but I feel like as a name or as a concept, maybe it requires a little bit of branding or a little bit of appreciation for what's going on. Make it kind of cool, and hopefully I'm doing that at least somewhat. But you have different factors here. Some of these microbes, they produce antimicrobial compounds, and it causes the botrytis to not be able to touch them. In other cases, they're parasites, and they damage the fungus. They give it a little taste of its own medicine, feels what it's like to be a parasite or to be parasitized. And so in this way, if you levy multiple factors that are very different, you're not going to have a selection pressure for your control, essentially. And of course, here's another example of a virus. Now, this is really interesting. These various strains of viruses were found in botrytis in China. And what was interesting was that what the viruses did, if I direct you to section C here, if you can see it, there's these little droplets. And they represent the botrytis. This is like a group of botrytis spores. But what was interesting was that when they had the virus, they weren't able to push through and penetrate into the cells. But if there was a wound, the botrytis was still able to infect the plant as normally. So the viruses, which are apparently really common, they didn't allow the botrytis to create these infection nodules that allow it to infect, but everything else was fine. So even when we rely on a biocontrol or another sort of aspect of the ecosystem to do its controls, it's only going to have certain weapons to do that with, and it might only affect one part of a large mosaic of effects. Like I said, this was pretty, I think that these viruses are rather ubiquitous, and this research showed that in a 900,000 square meter area in China, they found these viruses in various places, and that's where the research was conducted, and that's where they found all of these sort of botrytis-affecting viruses. I don't think there's a reason to think that these are not particularly rare, and they're probably found all over the world, but because of technology and our ability to understand them and find them, I think that we're dealing with something that's a lot more common, and I think, again, something that for us who are really interested in ecosystem health and dynamics, we should pay more attention to. So now we're going to talk about budworm moths, Helicoverpazea, and others in that group. We can see 3 life cycles here. The egg in the bottom right, the larva on the bud, and also this sort of nondescript moth, which if you didn't know better, you think might be nothing to worry about, but actually is a huge problem. So the corn earworm, perhaps unsurprisingly, is really common in North and South America because that's where corn is from, so that makes sense, right? But it actually feeds on many other kinds of plants: sorghum, cotton, beans, cucurbits, cabbages, all kinds. So they're actually a huge generalist, and they have a lot of adaptations that let them feed on these plants and not really deal with the fallout of their defenses. They've been tracked moving up to 750 kilometers as adults from Texas to Arkansas, and throughout the seasons, like in the United States, for example, you'll have populations that develop from overwintering in like the southwest, for example, and then they'll start their flight into the East Coast, and then they'll mate with the East Coast populations and hybridize, and so they will pass on and some of their defenses and other traits that allow them to be more effective, which is kind of interesting. A very common thing for budworm are BT, or Bacillus thuringiensis. Fun fact about BT, and this is not meant to scare anyone, but again, to talk about the, I guess, vivacity of the ecosystem, is that Bacillus thuringiensis is closely related to Bacillus anthracus, which produces anthrax, and also Bacillus cereus, which is a human pathogen. And again, that's not to say that BT is bad or anything like this, but I guess I'm trying to, again, belabor the point that we got good guys and bad guys that are very closely related, and maybe it only takes a few genes going one way or another for things to be different, and sometimes radically. But I bring this up because BT is effective, but it can also be resisted by the budworm moth. How does it work? Well, essentially, and again, if you want the presentation to see these diagrams a lot better, please ask me, because I think they deserve being seen. But essentially what happens is BT, as a bacteria, it produces these what are called protein crystals, and essentially they actually don't activate unless they're in an alkaline solution. Many caterpillars have an alkaline gut, and budworm is no exception. The reason for this, the reason why it's helpful for the caterpillar is because it actually neutralizes a lot of defenses just by the pH being radically different. So when they're feeding, some of these defenses and enzymes that they're ingesting, they just get neutralized by the pH, which is kind of fascinating. But unfortunately, the BT crystals need this, and so they change and they create this basically elaborate pore-producing structure. And then they basically put a bunch of needle points into their intestinal lining, and the caterpillar gets sepsis, basically. That's how it works. There's a bunch of other proteins. This one's modeling cry proteins, but there's a bunch of other ones as well: CYP, VIP, site, surface layer, perisporin, etc. And there's no reason to think that this is going to be a problem for us. We have a very different gut, and these crystals, these proteins, they only affect caterpillars, which is great. But when it comes to like adaptation to different plant hosts, this diagram is also kind of busy, but the first two rows are actually kind of intuitive. Lighter blue shows an increase in the expression of one of these 5 major detoxification gene families. So they have whole arrays of genes that are meant to combat, neutralize, or suppress defenses in plants. And I'm not going to get into the details about how they work, just know that we can see that different structures, different organs in the budworm moth and other caterpillars, they'll be differently expressed. But in particular, this is dealing with the budworm moth. So their legs, their antennae, their mouth parts, of course, a lot of these genes are highly expressed. But also, the bottom row shows different plants, and I don't think you can see it very well here, but we have geranium, tobacco, tomato, capsicum, cotton, Arabidopsis, which is thale cress, and maize. And we can see that if it's pink, or if it's a light pink color, then it's way more expressed, these different genes. And if it's green, then these groups were less expressed. And we can see that there's a big difference between, for example, tomato and maize versus tobacco and cotton. They were more expressed in one and less expressed in another. And so it's not enough to know that they can resist things like chemical compounds and other sorts of things. They're actually very variable and very mutable when it comes to different plant hosts. And I also want to say that they have a special organ called a fat body, not a fat cell. They do have those too, but fat bodies are special organs that insects have that are just totally for this detoxification effect. So I think a lot of people don't know the deep arcana of insect physiology, and I can't blame them. But yeah, there's a lot to it. I know, right? It is funny. So another thing I want to say, like I mentioned earlier, so this is cotton bollworm, which is another kind of budworm. So it's not the corn earworm that many people deal with in cannabis, but it's very closely related, and in some cases it's been found to hybridize between the two species, so I'm going to consider them to be effectively related and important to talk about. Cotton bollworm has a great gamut of chemical resistance abilities. Some of the major hits include things like, DDT, pyrethroids, spinosins, carbamates, and organophosphates, which are really terrible and a lot of places have outlawed, and you shouldn't use those on cannabis. But it just shows how effective. I mean, most people grew up knowing that DDT pretty much killed everything, but these caterpillars are here resisting it, which is pretty amazing in my opinion. And this table shows some of the modes of action of that resistance and some of those detoxification gene families I talked about earlier. Also, cannabis produces a lot of compounds that we appreciate and we care about. Of course, things like alpha and beta pinene, myrcene, borneol, methyl salicylate. These compounds are important for flavor and taste, and they're also important for defense. But they're also huge attractants of budworms too. So you kind of can't get away from it. And what's also been found to be the case is that for a lot of budworm moths, if they're raised on a certain plant as caterpillars, the adults will seek out those plants because they were raised on them, and so they've associated with that as a compatible host. This is obviously important for cannabis because a lot of people are dealing with budworm moths, and I wonder to what degree we have already gotten like specialist populations that might be trained or really heavily biased towards seeking out cannabis, especially in outdoor populations. Yeah, like I said here, loud is loud to budworm moth antennae, so it's something to consider, I think. And then we'll move on to aphids. Here's an example of a cannabis aphid on the right that has some russet mice on its body. That's kind of interesting. I have a video on my YouTube channel that goes over this in more detail and some of their other aspects. And then we have a lady beetle larva on the left for those who have never seen one before. And lady beetle larvae can eat from 50 to 200 aphids a day, depends on a lot of factors. And so, but here we have an aphid just walking across one, so they can't get everything, right? I've talked about it already, but the gut symbiont of insects is really, really sophisticated. It's what allows them to feed on their host oftentimes. For example, termites, which we're not worried about for the most part, they aren't able to eat wood without the bacteria in their gut that will allow them to digest it. And the same thing is true for things like aphids. They have many different things that allow them to subsist in the environment. On the right, though, we have an example of what's called deimatic behavior, which is not symbiont related, but it's another sort of factor. This is this movement in unison that they have, which startles. That's what deimos means in Greek. Things like parasitic wasps and other generalist predators that might be like, oh, I don't know what that is. I'm going to go away from it. And I guess it works out for them. I don't know. This is on a cannabis plant. I don't know the aphid species here. It doesn't quite look like a cannabis aphid, but I thought I would bring that up because it shows a weird behavior that people might not be familiar with. But some of these symbionts I was talking about, they do things like provide essential amino acids. They provide defenses against parasitoids and pesticides, and we'll go into some of those examples here. Like Tyler had mentioned earlier, it's been shown in research that aphids actually, they really need the plant sugars. The sucrose in a lot of plants is actually a phagostimulant, so it actually stimulates their feeding response, and it's like the number one thing that they are kind of as a gustatory cue when they're feeding and going, is this a host that I can feed on? Most aphids are specialists, and so they can only feed on one or a few different host species. And so, but that sugar content is actually very important for them to tell, is this something that I want to eat? And they have a lot of enzymes, as I've highlighted here in the top, that they use to break down the sugars and digest them. As an example, one research report looked at like how much sugar can an aphid feed on, and in a solution where there's 1 mole of sucrose in 1,000 grams of water, they found that basically they had no problem feeding on what essentially amounted to, if my math is right here, about 25 degrees Brix, or 25% sucrose by max. Obviously, there's other aspects of the the phloem sap. There's not just sugar in there, of course, but it's these sugars that are often depicted as important for aphid feeding. And according to my research, at least in 2021, they do sometimes change the formula, right? But Coca-Cola, a 2-liter Coca-Cola, only has 11.7 Brix, which is interesting to note. So they have quite the sweet tooth. And they deposit honeydew, which is also sugary and attracts things like ants. A lot of people here are aware of that. But what you might not know is that aphids can actually affect the soil microbiome 2 ways. The first way is by producing the honeydew, and that honeydew falls off onto the leaves and the foliage and also into the soil. And by doing that, it increases the carbon content quite a bit. And what's been found is that this might actually have a negative effect by increasing microbial competition with the plant host for those carbon resources, because they're still going to be, in some cases, nitrogen limited. And so that causes this sort of like massive competition effect, which is really interesting to consider. And also on the foliage too, it will change the phylosphere dynamic of the microbiome. So all the different microbes, the bacteria, the fungi, etc., that are on the leaves, that can change pretty rapidly. They looked at 3 different soils: a clayey soil, I don't know about that terminology, mixed soil, and sandy soil. And they found that the aphids were affiliated with different kinds of microbes depending on the kind of soil as well. So I think that's a really important thing. It's something I didn't expect. And then I guess there's a third way that aphids affect the microbiome, which is by suppressing the immune system of the plant cell, or sorry, not the plant cell, but of the plant. And as they reproduce, you get more and more of this stacking effect. So usually it's local, but it can globally affect the plant as well as the population gets larger and larger. Oh, I didn't get that text, unfortunately. But we can see here that aphids will shape the microbiome, and they may create a benefit to themselves, or at least a detriment to the plant. And this diagram shows that in comparison to the control here, places where black city mold which is where honeydew has fallen, because we have these city molds that will colonize on honeydew. That carbon level was massively more, like maybe 5 times as much or something like this. So that will definitely have an effect, and it will change the dynamics of the microbiome. Aphids will also change color a lot of the time. This is a pea aphid, and their symbionts can affect this, but also their genes can, and their level of starvation can also affect pigment synthesis, which has a lot of interesting effects that will shape whether they interact with biocontrols the same way, but it will also change their resistance to things like, UV spectrum outside, or if you're using it as a way to control them. It will affect their fecundity and their longevity, and a bunch of other factors too, which I think is interesting. If they're starving, like an example, the pea aphid is a model aphid that we use to research aphids generally. It was found here that if they are starved, then they will change their coloration. But what's also been found is that a microbe called Rickettsia viridis like green, will cause the pea aphid to be a green color. If it lacks Rickettsia, then it will be red, and lady beetles, many of which are red colored, will be able to find it on the green leaf background and feed on them. And so that has a bias to the population, and it will select for maybe those that have the green and the Rickettsia. However, pea aphids that have Rickettsia, they are actually more attractive, or at least they can be found easier by the parasitoid wasps. So then they have the selection back towards the red coloration. So you see this dynamic can actually play out. And for other aphids, similar things are also true. And then this is my favorite example here. There's what's called a hypersymbiont, a symbiont of symbionts, in a bacteria found in many aphids called Hamiltonella defensa. It's appropriately named. Basically, this is a bacteriophage, which is a type of virus that normally colonizes and kills bacteria. And we'll talk about those a little bit more. This is a lysogenic virus. That basically means that its genetic code has been integrated into the genome of the bacteria. So it doesn't exist as its own separate thing. But what it does contribute to the bacteria is that it contributes these little toxins. These toxins kill a larval parasitoid wasp as it's developing in the aphid. I've had many people come across me and ask, hey, what do you think of a parasitoid wasp? Or does this work? I've used them before. They don't really work very well. This might be one reason it didn't work very well. And it's very hard to tell this is happening in the field, of course, but it's a factor nonetheless. And what we also find, and that's a picture of a bacteriophage of the same kind of group. That's what they might look like if they were not in the genome. But there's also different strains, right? And so different strains of the bacteria itself, different strains of the aphid itself, and also different strains of the virus itself. APSC, which stands for Acyrthocyphum pisum, pea aphid, secondary endosymbiont, APSE. So there's different strains of this virus, and so they'll cause different kinds of reactions, and they'll give different levels of protection against the parasitized wasp. I just think this is really fascinating, and I look forward to the future, maybe the near future, when we're able to quantify this information to our own benefit. Another thing we can do to disrupt the ever-swarm, so to speak, is to not just send out some biocontrols like wasps and things and not provision them with resources. For example, here we found that if you just let them be starved, these parasitic wasps, they didn't last very long. But if you gave them honeydew or a sugar substitute, or you gave them water, they lasted a little bit longer. If you gave them both, they lasted a lot longer. And so people have asked about using banker plants and things like that. And with the right setup, I think it can be really salubrious to your IPM strategy. Here's another hyper symbiont, so to speak, that we could talk about, which is a hyperparasitoid. So if you've ever used parasitic wasp and they haven't worked, this might be another reason, especially outdoors, where they might not have worked well. It's been found that there are parasitic wasps that will go and search aphids that have parasitic wasps, and they colonize those wasps. I know it's a big tongue twister, but this can actually be a major downturn for populations of parasitic wasps in the field. And so what's been found is that if you increase the amount of banker plants, if you increase the amount of parasitic wasps in the area, you might also increase these hyper parasitic wasps, right? And it's hard to tell. They look very similar. You won't be able to tell them with your eyes, for example. But they were able to collect them in this example, and they were able to show that that was the case. And then finally, aphids are the biggest vector of plant viruses. Oh, I am a little bit over time. Sorry about that. But they are the biggest vector of plant viruses of all insects, and they've co-evolved with viruses quite a bit. Some aphids allow viruses to do better, and some viruses allow aphids to do better. And depending on how consistent the plant host is, as well as the aphid host is, they might develop a co-evolutionary interaction whereby the virus either makes things a lot easier for the aphid, or, ironically, are you the cane?
Please, finish. We can finish. We need to
finish. Okay. Yeah, let me finish quickly. Long story short, basically, we should respect the ecosystem and the various minutiae that we're dealt with. If the virus population is consistent, then you get situations where the virus actually makes the aphid maybe not like the plant, and it will taste the plant and it will go, ooh, this is gross, and it will leave. But the virus still gets vectored. And here's an example of aphid suppression. These GIF images were a lot for the presentation to work with. So basically, if you see red, that's the calcium influx that shows immune signaling. And in some plants, we can bioprime them to have a greater reaction, and so they won't be suppressed by the plant's various ways of suppressing the plant immune system, which we'll go into. And there's two phenotypes for aphid virus interaction. One is attract and deter, which I mentioned earlier, and one is for retainment, and that will change how viruses move in a crop. And then this is to scare you a little bit. These are a bunch of bacteriophages found on leaf. Each one of these color-coded globules show different bacteriophages that affect bacteria. And then I'll just give you this example here. In the soil, there's a lot of them. I'm going to go through really quickly.
We got to stop sometime.
All right, this is the last slide. Viruses that affect moth larvae are everywhere, and they're also in the soil, and the living soil should consider that. Thank you very much. Sorry for taking your time.
Great job. How could I have stopped that? Jeez, man, I'm the biggest a-hole here for stopping that and slowing that down.